Bismillah ar-Rahman ar-Rahim. We are going to start the course of the pharmaceutical microbiology. And the first part will be with me, Dr. Shireen Gamal El-Gindi, Assistant Professor of Medical Microbiology and Immunology, Faculty of Medicine, Asyut University. Objectives. At the end of this lecture, you should know some important definitions concerning sterilization and disinfection, reasons to learn sterilization and disinfection techniques, the kinetics of sterilization and disinfection. Also, you should know some important expressions of resistance, methods of sterilization and disinfection, and finally, sterilization indicators. To describe the damaging effect of certain chemical or physical agent on the living microorganisms, many terms are used. Sterilization. It is a total kill of all living microorganisms, including viruses, fungi, bacteria, and their spores. Spores are a very resistant form of bacteria, and sterilization kill these spores. Disinfection is the killing or removal of the harmful vegetative microorganisms, but not include the spores. Disinfectant is a chemical substance that's used to kill these pathogenic microorganisms on non-living objects. For example, from the surgical instruments, from the monitors, from the floor, from the benches, from non-living objects. But when this disinfectant can be used safely on living tissue, it's called antiseptics. Okay, why we study the sterilization and disinfection techniques? Sterilization is required in the following fields. In the field of microbiology, safety in the lab requires sterile or aseptic techniques. Also, the accuracy of microbiological tests require completely sterile equipment. In the field of pharmacy, we can't imagine all the pharmaceutical products present in the pharmacy, including injectables, ophthalmics, and other products. We can't imagine it non-sterile. In the field of medicine, we can't do any operation without the sterilization of the operating room, without the sterilization of all instruments used in this operation. And so sterilization and disinfection is very, very important in all these fields. The order of resistance of the most known microorganisms, the hardest organisms to kill, are the prions and the spores. They are very, very resistant. And the easiest to kill are the vegetative bacteria and the enveloped viruses. Enveloped viruses, for example, the COVID-19 virus, that can be easily destroyed by the alcohol. Kinetics of sterilization and disinfection. To describe the sterilization effect of certain lethal agent on certain number of the microorganisms by time, the disk curve is obtained. This disk curve shows the number of survivals by time. This number of survivals, as shown in the figure, shows steady decrease by time when it is subject to certain lethal agent. Again, when certain number of bacterial cells is exposed to lethal agent, there is a steady decrease in the number of survivors with time. This steady decrease is called exponential kinetics, exponential or logarithmic kinetics. So the normal death curve is exponential or logarithmic. But sometimes deviation from linearity is occur in the death curve. As appeared in the area 1, it may give initial hump or maybe give straight tailing or straight line as in the area 3. The first part, which is appear as an initial hump, this is due to the heat activation of spores. That means if the organism is exposed to the lethal agent containing spores, when this spores is exposed to heat, firstly it is converted to the vegetative form, and so the number of bacteria is increased and giving initial hump. After that, and by time, it will give the steady state decrease again, which is the area number 2. Sometimes the area number 3 or this straight line or tailing is appear due to the presence of very heat resistant bacteria. There are different methods to achieve the sterilization: physical methods or chemical methods. Physical methods including heat, radiation, filtration, and chemical methods including liquids and gases. The choice of the probable sterilization process is very important to prevent the product deterioration. That means, for example, when I want to sterile the plastic syringe, I can't use heat as a method for sterilization. This is because the plastic syringe will be melted by the effect of heat, and so it is deteriorated. Another example, when I want to sterile the antibiotics or any other biological fluids, I can't use the heat as a method for sterilization. This is due to the inactivation of these antibiotics. Okay, sterilization will occur, but the product will be deteriorated, will be inactive. So the success of sterilization process depends upon suitable choice of the method that is probable for my product. I want to obtain maximal microbial removal and minimal product deterioration. Some important expressions of resistance you should know. Thermal dew point. Thermal dew point is the lowest temperature required to kill all the microorganisms present in this product within a certain time. Lowest temperature required to kill the microorganism and to achieve the sterilization. While thermal dew time, it is the shortest time required to kill the microorganisms present in our product at certain temperature. Very important to determine the thermal dew point and thermal dew time, as I want to obtain the lowest temperature required to achieve the sterilization process. I can make much more rise in the temperature, but why? It will cause damage to our product. As in case of antibiotic, we can use a large doses of antibiotic, much more than the MIC or the minimal inhibitory concentration. I can use much more larger doses, but I will obtain the larger side effect. So I use the lowest concentration that give inhibition of the microorganisms in case of antibiotics. Here also in the non-specific antimicrobial agent and in sterilization processes, I want to obtain the lowest temperature that achieves the sterilization process and also the shortest time. Another expression is the decimal reduction time or D value. This is the time at fixed temperature required to reduce the number of the viable cells by 90%, and it is much more applicable to reduce the number of viable organisms by 90%. This is because the presence of heat-resistant bacteria, as we said before, in the deviation from this curve. Again, the value is the time at fixed temperature required to reduce the number of the viable cells by 90%. Here we measure the degree of resistance of microorganism at particular temperature. Z value, the increase in the temperature required to reduce the D value by 90%. Here we change the temperature in order to obtain reduction in the D value. And so we measure here the change in resistance of the microorganisms upon change in the temperature. Another expression of resistance, inactivation factor. What is the meaning of inactivation factor? Inactivation factor, it is to measure the total destructive power of sterilization process. In the field of industry, when I obtain any batch of raw materials or other, I want to determine the initial number of the microorganism before I accept it. And the number of the microorganism must be within the numbers described in the pharmacopoeias. I can't take the raw materials that is completely contaminated with the microorganisms. It will be very difficult to be used. The initial number of the microorganisms before any sterilization process is called the bioburden. And after sterilization process, I count the microorganism again, and by division of the initial number of the microorganism before sterilization over the number of the microorganisms after sterilization in our raw material or any other pharmaceutical product, it will give the inactivation factor, which is the total destructive power of sterilization process, and it's called IF. Again, the IF is equal A over B. A is the bioburden and B is the number of microorganisms after sterilization. The highest is the IF, the more the efficient sterilization process, and it is described in detail in the pharmacopoeias, and the maximum allowed IF value is 10 over 7. Higuchi-Ampas equation. To describe the effect of heat and extension time, Higuchi-Ampas equation is presented as it appears from the figure. The extension time is inversely proportional to the temperature. That means any small increase in the temperature greatly reduces the time required to kill the microorganisms and time required for sterilization. The extension time is inversely proportional to the temperature, and it is very clear from the figure. Now we are going to talk about the methods of sterilization, but this part will be discussed in detail in the practical section, and so we can take it shortly. Physical and chemical methods. Physical methods including heat, radiation, filtration, and chemical methods including liquids and gases. And in general, you should know that most of the chemical methods achieve only disinfection, not reach to the level of sterilization, but there are certain exceptions. You must know it. Physical methods are heat, radiation, and filtration, and all achieve complete sterilization. We will begin talking with heat. There are two types of heat: moist heat and dry heat. moist heat and dry heat. When we compare between the moist heat and the dry heat, the mechanism of action of this heat on the microorganism, dry heat makes certain oxidation of certain proteins present in the bacteria, followed by extensive dryness, while the moist heat causes coagulation of the total protein of bacteria. The time required to achieve the sterilization by dry heat is longer than the moist heat. The temperature required is higher in dry heat than in the moist heat. The damaging effect on the article is higher in dry heat than the moist heat. And finally, penetration power of dry heat is lower than the moist heat. So in comparison of dry heat with the moist heat, we found that moist heat is much more efficient than dry heat. But dry heat also have many uses, as we will discuss now. Dry heat, direct heat. Here, the object is directly placed in the flame or in the Bunsen flame, as in the figure, in the sterilization of the bacteriological loop until redness of the loop to achieve the sterilization. This is called red heat. Also, flaming of the mouth of tubes and glasses is a type of dry heat. Also, incineration. Incineration is required in certain cases. All the hospital waste that are very and seriously infectious agents, all the contaminated materials that is considered the infectious hospital waste must be completely incinerated. Also, certain bodies of dead animals infected with serious infections as anthrax that containing the spores must be also incinerated. Hot air oven, as it's clear in the figure, it is a double-walled metal chamber containing trays and perforated from inside. From these pores, hot and dry air is entered and circulate inside in order to sterile all the equipment that I will put inside. It is very simple. The temperature here in the hot air oven is 160 degrees for 2 hours or 170 degrees for 1 hour. This temperature and time is very, very important to achieve the complete sterilization. The hot air oven is used for sterilization of the following. Very important to know each apparatus or each technique and its usage. As we said before, the choice of sterilization process, our appropriate sterilization process, is very, very important. Here, hot air oven can be used for sterilization of all the glasswares, talc powder, surgical instruments, sand, the swabs, sulfa drugs. Sulfa drugs is the exception of any antimicrobial agent that can be sterilized by heat. And finally, any pharmaceutical product containing fat, waxes, or oil, and Vaseline. All these products and equipments can be sterilized in the hot air oven. Sometimes our product not withstand this heat for this time, and fractionation of this dry heat is necessary. As in case of serum and egg containing media, the fractionation of dry heat by the hot air oven and the temperature is reached 80 degrees for 1 hour on 2 successive days. Fractionation of dry heat by the oven at the temperature 80 degrees for 1 hour on 2 successive days is called inspection. And the inspection is used for the sterilization of serum and egg containing media. And the final technique in the dry heat is the infrared radiation. Infrared radiation is obtained from infrared radiation lamps. Here, the infrared radiation is used as a source of heat. It gives heat reach to 180 degrees, and for 20 minutes it achieves complete sterilization. But it has a limited use due to its carcinogenic effect, and they use mainly for the depyrogenation of empty ampoules and vials. Again, the infrared radiation is considered as a method of heat for sterilization, not a method of radiation. Why? Because we use the heat obtained from the IR radiation as a method of sterilization. We use the heat as the tool of sterilization. This is a tunnel that contains the infrared radiation lamp inside, and the product enter inside the tunnel, and it's exposed to the heat, which is 180 degrees, and for 20 minutes it will give complete sterilization of the products. Now we are going to talk about the moist heat. Moist heat at temperature below 100 degrees, as in case of pasteurization of milk, preparation of heat-killed vaccine, and tendilization of the serum. Pasteurization of milk. Louis Pasteur demonstrates the method for sterilization of milk. This process depends mainly on the principle of heat shock. The milk is heated to 63 degrees or 72 degrees for a while, then rapid cooling below 10 degrees. So the pasteurization of milk is depending on the heating, then sudden cooling. This makes heat shock for the bacteria and death of the bacteria as a Holder method or flash method. Okay, now 63 degrees or 72 degrees is efficient for sterilization. Sterilization, that means killing of the bacteria and its spores. No, it kills only the vegetative form of the bacteria, but if you know that most of the organisms come from the milk or infect human from the animal source through the milk, as Mycobacterium tuberculosis, Procella abortus, Salmonella, all these organisms are non-spore forming organisms. So the pasteurization of milk is efficient for sterilization of milk. Preparation of heat-killed vaccine at 60 degrees for 1 hour. This preparation will be discussed in detail in the practical section. Moist heat at 100 degrees. It is a boiling for 5 to 10 minutes in water. The boiling of the instrument in water for 10 minutes is sufficient to kill all the vegetative bacteria and even the hepatitis viruses. But this method not used. Only use this method if there is no other available method is obtained. If no other method is present, we can use the boiling for 10 minutes in a hot water at 100 degrees. Steaming at 100 degrees in Koch steamer. This method is used for sterilization of some culture media that's containing sugar and gelatin, but the Koch steamer require 100 degree for 90 minutes. 100 degree for 90 minutes. The Koch steamer is simply composed of two pans inside each other. The inner pan is perforated, and between the two pans there is a water and heat coil. When the heat coil is begin to heat, the water will boil and produce its steam. The steam enters through inside the pores of the inner pan, and so the product that want to be sterilized will be sterile. The Koch steamer, to obtain complete sterilization in Koch steamer, we require 90 minutes in the Koch steamer to obtain this sterilization. But sometimes the preparation don't withstand 100 degree for 90 minutes, as in case of sugar and gelatin culture media. And in this case, we fractionate this moist heat at 100 degree for 30 minutes on 3 successive days. So sterilization by tyndallization. Tyndallization is the fractionation of moist heat at 100 degree for 30 minutes on 3 successive days. This is in case of the sugar and gelatin containing media, and this is achieve complete sterilization and killing of even the spores that vegetate in the first and the second days.